look at that flower. Look how beautiful it is, said an artist to his friend. The artist said that science just takes apart that flower and makes it dull. But the friend happened to be Richard Feynman, the Nobel Prize winning physicist, and he thought that this artist's view was a bit nutty. He thought, well, as a scientist, I too could see the beauty of that flower, but as a scientist, I can dig into why the flower has those colors, what the molecular processes are inside the cells of the petal that give the flower that color. And more than that, I can think about why the flower has those colors, which is, of course, to attract insects, which suggests something about insects having a sense of aesthetics and brings up all kinds of issues about how evolution works. So as a scientist, I don't see that flower in any duller way. In fact, science only adds to the wonder and beauty of that flower. Richard Feynman said that in 1981 on BBC TV. Rose is swaying behind him, and I was 11 at the time, and he said it better than I could have said myself. Science only adds. I already knew I wanted to be a scientist, but he's just said it so perfectly. The insides of the human body are not aesthetically pleasing like a flower, but wonder and beauty ascends from the details of how the body works. And the part of the human body which we understand the most is the immune system. What happens when you get a cut, when you get that sort of tender redness or swelling and inflammation? That process is what we understand the best in everything about how the body works. Cells move into the region where you've had a cut, a choreography of events, different kind of cells move in there to clear out any opportunistic germs and to begin the healing process. That is the part of the human body which we understand the best. And from all that understanding, I don't think it's an overstatement to say that we're at the cusp of a health revolution in the new kinds of medicines we can create from all the knowledge we have about how the immune system works. So in today's talk, I want to talk to you a little bit about how we get to this revolution. I want to give you a sense of the discoveries that have happened over the last few decades that have got us to this revolutionary moment in our understanding of the immune system. And then I want to go through some specific examples of how this directly leads to new treatments for autoimmune disease, new treatments for cancer, and I want to get across a sense of where that's going in the future with a little bit of my own research at the end. So I'm going to begin with the revolution in immunity. And we're going to start with Charles Janeway. Any starting point in a revolution is a bit arbitrary, but we'll start with Charlie Janeway. He had an epiphany in 1989. He was thinking about two particular things. He was thinking about how vaccines work and that there was a crucial problem in our understanding of how vaccines work. As you know, roughly speaking, a vaccine works by the fact that you're exposed to a dead or harmless version of the germ. The cells in your body that recognize that germ multiply in number. And then if you're exposed to the real version of the germ, you would have cells readily there, because some of them live in your body for a long time, to react against the real germ. Roughly speaking, we all understand that. That's how a vaccine works. The gap in the knowledge was this. It was that if you took an animal, a mouse, and you gave it a bacteria, then a dead version of the bacteria, then it would make an immune response. If it saw the real bacteria, it would then be able to deal with it. The vaccine would work. But if you gave the mouse one isolated protein molecule from the bacteria, it still is not from the mouse, it's from the germ. The mouse should see that as a foreign thing, as something from bacteria, and it should mount an immune response, but it doesn't. It doesn't respond to just a small protein molecule, one type of protein molecule isolated from the germ. It only would respond to the whole germ. And people thought, people thought that there was something strange about that, but they didn't really seize on it as a major gap in our knowledge. It happened by chance that it was discovered that if you mix up the protein molecule with aluminium salt, then it would work as a vaccine. And this small technical tip didn't quite make sense, and Charlie Janeway seized on that as a major gap in our understanding. There was something mysterious about what a vaccine, what makes a good vaccine. The other thing he was thinking about was he was at a scientific meeting with his wife, Kim Bottomley, also a famous immunologist, and they were listening to the talks, and all the talks were about how vaccines work, as in what makes these cells in your body live for a long time so that you could respond better the second time round. The crucial thing they weren't talking about was what triggers an immune response in the first place. And he said to him and his wife were discussing that. We don't really understand what triggers an immune response. He thought about it for 6 months. He put those two things together and he had his epiphany, which was that we don't really understand, this was in 1989, what triggers an immune response. He postulated that there had to be a whole undiscovered part of the immune system that was directly able to recognize germs. It can't be that your body responds to anything that's not part of you. And now, in hindsight, that obviously makes sense. Your body doesn't want to respond to the food you eat or the dust in the air. Your body has to specifically respond to germs that might cause a problem. And Charlie Janeway stood in 1989 in front of all the world's best immunologists and said, look, there's a gap in your understanding, and it's this, that there must be a part of the immune system that can specifically detect germs. He postulated that there had to be receptors that would protrude out from immune systems and lock on some way to detect bits of germs. There was no evidence for his idea. He published it, and it was pretty much ignored for 7 years. It touched this person 4,500 miles away, Rizla Mezatov. He was born in Tashkent, Uzbekistan, but he was a student in Moscow University. And he, as a student, as an undergraduate student, he wasn't allowed into the library that held this paper that Charlie Janeway wrote, but he charmed his way into the library. He spent a month's stipend photocopying the paper, and he said it was a visceral light bulb moment, that it had to make sense. There had to be a part of the immune system that we hadn't discovered yet that had to be able to directly sense germs. He started emailing Charlie Janeway to discuss the idea. It was the time that the Soviet Union was breaking up. There wasn't a lot of money for scientific research. The Moscow University was allowed to send 300 words out by email. And so Rizla Mezatov charmed his way with the librarian to get that to work for him. He wrote what he wanted to say, gave it in a floppy disk. They would then send this out to Charlie Janeway. Charlie Janeway was quite excited by this because his idea would have been ignored, and he thought, oh, this is great, this guy's corresponding with me, so he was quite pleased about that. Eventually, Rizla Mezatov asked if he could come and work with Charlie Janeway in Yale. Charlie Janeway discussed it with his wife, Kim Bottomley. They thought, well, we don't really know much about this guy in Moscow University. Maybe not. So Rizla Mezatov, though, won a fellowship for 3 months to go and work in the USA. And he was working on analyzing genes with computer software, which was a new thing at the time. And sometime soon before he had to return back to the Soviet Union, he gave a talk. And in the talk, in the audience, was Richard Dutton, the head of the American Association of Immunology. And after the talk, Richard Dutton spoke with Rizla Mezatov. He said, oh, your talk was very, very good. It's very interesting. And Rizla Mezatov explained, yeah, I've got to go back to the Soviet Union, but I'd really like to work with this guy in Yale, Charlie Janeway. And Richard Dutton then sent an email to Charlie Janeway saying, oh, I've just heard this guy speak. Actually, I think he's actually pretty good. And then the next morning, Rizla Mezatov would offer the job. He moved over, and finally, Charlie Janeway and Rizla Mezatov met. But how were they going to solve this problem of whether or not the body had some previously unknown way of directly recognizing germs? Rizla Mezatov was not an experienced experimental biologist, and the problem is pretty hard. We're now going to shift to Paris, where the next part of this story happens. This is Jules Hoffman with a PhD student of his that became his wife, Danielle Hoffman, and his dad was an entomologist, and he was thinking about insects. He reasoned that 80% of the world's species are insects, and insects are also attacked by bacteria, viruses, and fungi. Insects must have some important antimicrobial defense. So this does not look like the path to a Nobel Prize, right? Thinking about how insects are not getting sick from germs. But it turns out that this is hugely important. He's had his lab starting off working on grasshoppers, but half the lab, including Danielle, who's going to become his wife, became allergic to grasshoppers. So he shifted the thinking, oh yeah, well, grasshoppers aren't so good. We'll work on Drosophila fruit flies. This is the experiment that he did to win a Nobel Prize. He essentially, with his lab team, he essentially pricked the fly with a bacteria or fungi, isolated blood from the fly, and then showed that the blood outside the fly, then in a dish, could kill the bacteria or fungi that the insects were exposed to. So that demonstrated that there was a strong inducible immune defense in a fly to kill off these germs. How is this triggered? Well, it turns out lots of different labs are working on these Drosophila fruit flies, so they could collect from other labs various genetically modified flies. And then they found one where they pricked the fly with a fungi, and then the blood from that fly now wasn't able to kill fungi in a lab dish. And that meant that the gene that was mutated in that fly was crucial to the fly's immune response. And that became called the toll gene. And this is the dead fly, overrun with fungus. It's really that fly that got the Nobel Prize. So that led to the discovery that there was a gene essential in insects that were enabling it to kill off, in this case, a fungus. But it still doesn't quite make total sense yet. Enter Bruce Beutler in Chicago. And he was studying a mouse. As many of you know, if you are given an acute bacterial infection, then the immune response against against the bacterial infection can sometimes be harmful, and that is called sepsis. If it becomes extremely harmful, it can be called septic shock. But he was studying a particular mouse that did not get sepsis. He knew that there was something about that mouse that it could not respond to bacteria like a normal mouse, and he realized that if you could work out why that mouse wasn't responding to bacteria, it would give you a good clue as to how the immune system worked. He says that the problem, though, was incredibly frustrating. He likens it to sort of looking for a coin in your front room. You just really never know when it's going to show up. And in fact, very shortly before he did discover what was wrong with that mouse, his funding agency said, look, you've been looking for this for ages, and your funding's we're done with that. But he did solve the problem. He found the genetic mutation in the mouse that made it not see bacteria. And the gene in the mouse that was essential for it seeing bacteria was similar to the gene in flies that was important for it seeing germs. And Rizla Mezatov and Charlie Janeway found a version of that same gene in us, in people. And so that leads us now to that Charlie Janeway's original idea was right, that we do have receptors protruding from our immune system that directly lock on to telltale signs of germs. This is the receptor that protrudes out from the surface of immune cells, and it locks onto, in this case, the outside bit of a bacteria, and that tells your immune system that you need to do something about this. It's not just that you respond to anything that's not part of you. You respond to things that are not part of you and are dangerous, like a bacteria. The impact of this directly is that it leads to the idea of adjuvants. The vaccine works not just by exposing you to something that's never been in your body before, but exposing you to something that's never been in your body before and will trigger an immune response, like a bit of a bacteria. Pharmaceutical companies saw this as a way to make money from vaccines, which kick-started a much bigger, broader interest in making vaccines commercially. For example, the papillomavirus vaccine benefited from this work. Crucially, the whole mission of the immune system changed with these discoveries, because it's not just about looking for things that are not part of you, it's looking for things that are not part of you and dangerous. And this opened up the complexity of the immune system, which means that it's not just a simple cell type in your body that fights off germs. There's a whole labyrinth, a world of different kinds of cells in your body, of all the different genes, proteins. It's been estimated that something like a third of the genes in you might be to do with your immune system to protect you against disease. The complexity of the immune system is the way now in which we can start to realize new medicines. And I'm going to go through a couple of examples of that. One of the things that directly comes out of the complexity in the immune system is that the different kinds of immune cells have to communicate with each other to coordinate their responses. One of the ways they do this is they send out soluble secretions. They send out soluble secretions, for example, in autoimmune disease, where you have an unwanted immune response that you want to switch off. Two scientists came up with the idea that you would be able to block the communication between immune cells by blocking those soluble secretions moving from one cell to another, and perhaps dampen the problem in an autoimmune disease. And that led directly to what's called anti-TNF therapy, an antibody that blocks one of the soluble secretions, TNF, that's important in autoimmune disease, in rheumatoid arthritis. And so you prevent immune cells egging each other on, and you dampen the problem in that particular type of autoimmune disease. Mark Feldman had the idea while he was on holiday in Spain. He says that we have to give everybody much bigger holidays to have big ideas. I can tell you that now because my lab's not here, right? So, and of course he worked with Tiny Maney, the clinician, to realize this, which is of course now a multi-billion dollar drug, which may or may not be important. What is important is a lot more people don't have to use a wheelchair because of this idea. More recently, the Nobel Prize at the end of last year was given for a new type of immune therapy against cancer, which is that, again, it came from curiosity-driven research. It was just that there were receptors on the surface of immune cells, and several labs, including Jim Allison, Tasuku Honju, and Jeff Bluestone and others, were investigating what do these receptors do on the surface of immune cells, and it turns out that they were important for switching off the immune response, which again, in hindsight, makes sense, but it wasn't so clear at the time, and that's because if you're infected with a flu virus, the cells in you that would kill off that flu virus multiply in number, but once you've eradicated that flu virus, for example, then you don't want all those cells still in your body, so your body has to switch off the immune response after some time to bring your body back to its normal resting level. But if it's switching itself off after some time while you've got cancer, then you don't want to switch off your immune response against cancer. And they had the idea that if you block some of these brake receptors on immune cells, you might be able to have a longer-lasting, better response against, for example, cancer. So there are brake receptors on immune cells, which are important to bring our immune system back to its normal resting state. But if those brakes come on against cancer, that might not be beneficial. They developed then antibodies that blocked these brake receptors on immune cells and got a better response in some types of cancer for some patients. This is the tip of the iceberg. That's why I don't think it's an overstatement for me to say that really this has just been a proof of principle, and there are, at the moment, there are, I think, 6 antibodies licensed, and they block 2 particular brake receptors. But there are about 20 different brake receptors in the immune system. They work on different cells, they work in different organs, they work in different situations. We can block each one of those brakes in turn or in combination with other drugs. And it's really, we're just at the tip of the iceberg in being able to nudge the power of the immune system up or down by blocking brakes or blocking co-stimulatory receptors or other approaches to tweak the power of the immune system to better enable it to fight off diseases like cancer or to dampen its activity in autoimmune disease, for example. They list all this work leads to sort of celebrity status of the people who are doing the work. I have to say that really everybody is standing together in the community, though. The work that I just presented, there are heroes. This is Jim Allison playing his harmonica at the House of Blues. But his work comes from the whole community of scientists, of immunologists, which there's tens of thousands of people around the globe digging into the details of how the immune system works. And it's from those details that we get new medicines. And the individuals are deservedly celebrated, but they stand as part of a community. This is Jim Allison meeting Willie Nelson. This is Jim Allison playing in the House of Blues, meeting Sean Parker, who set up the Parker Institute that funds a lot of the exciting new ways of boosting the immune system. And here's Jim Allison's wife, Padmini Sharma, as well, meeting the Edge from U2. But it's the next photo that he and the whole community is most proud of. This is Jim Allison with Sharon Belvin, who was initially given 6 months to live and is now still alive much more than a decade later because of the antibody drug that Jim Allison first used to show that this would be a real treatment. Sharon Belvin kept in touch with Jim Allison a couple of years after her treatment. She had a child. A couple of years later, I think she had a second child. And both the Talmud and the Quran, of course, say that anyone who has saved one person's life has saved the whole world. My own work, just to give you this is a huge story spanning the globe, spanning tens of thousands of people, really, over decades. And that's what my message is, that this is a massive revolution that's coming over some period of time. But of course, I'll just give you a glimpse of my own research as an example of where things are going and how lots of people are digging into details and new ideas. We use super resolution microscopes. They won the Nobel Prize in 2014. And we dig into how immune cells recognize whether other cells are healthy or diseased, and then when they see a diseased cell, how they execute that cell or secrete molecules to trigger other immune cells. So this would be a natural killer cell, one of your white blood cells, and it's seeing this other cell. It's going to decide if that other cell is healthy or diseased. If it's a cancer cell, it would send those red packets of toxic proteins across to the contact between the cells and kill the diseased cell. This is our latest. Scientists are always very excited about their latest discovery. This is our latest discovery. One of the things that happens is that when an immune cell sees a disease cell and then it would kill it, then the receptors at the surface of the immune cell, they get chopped off. And people thought that those receptors must get chopped off perhaps as a part of dampening down the immune response. We've already spoken how after time your body needs to come back to its normal resting state. It has to switch off. And this process of chopping off the receptors that would see the disease cell must be part of dampening down the immune response. And there were some ideas about how you might want to stop chopping off the receptors so that you would boost your immune response to better kill disease cells. But as it turns out, just by using these microscopes to watch what happens, actually chopping off these receptors doesn't dampen the immune response, it boosts the immune response. And why is that? That's because you need to chop off the receptors that would detect the disease cell so that the immune cell can detach itself from the disease cell that it's just killed and then go and kill another cell. So when an immune cell comes up against a cancer cell, it will then see it as a cancer cell, it would then kill it, and then it has to chop off those receptors that see that disease cell so that it could detach itself and then move on to go and look at another cell, which means that chopping off these receptors might be important for boosting the immune response rather than dampening it, and you don't want to prevent that process. And this is, of course, a good example where now we've got a sort of another window. So you start to see another side of the process by which the immune system works, and you and I could actually easily think of ways in which you might be able to look at this kind of process and think about new ideas for medicines that would boost not just the way in which the immune cell detects a diseased cell, but its ability to then detach and then go and kill a second immune cell to efficiently kill off cancer cells. Okay, this was very short. I wanted to get across a sense of this decades-long journey involving tens of thousands of people, myself just part of an enormous community, where there has been a revolutionary understanding of how the immune system works, which I believe is the bit of the human body that we understand the best. This is directly leading to new medicines. There's already been good examples in autoimmune disease and in cancer, and there are lots and lots of big new ideas out there about how we might tweak the power of our immune system in different ways to deal with different diseases, and this is going to be ever increasing the number of ways in which we could tweak the immune system. Of course, all of everything I've said is important because of the medical discoveries that we get from understanding the immune system. But I have to say that personally, as a scientist, it isn't really the medical breakthroughs that get me to do the work. Really, it's all curiosity driven. And most of the discoveries that I spoke about today came from curiosity driven research. Jim Allison and Tasuku Honjo did not deliberately set out to cure cancer. They set out to understand what this particular protein molecule did in how the immune system works, and they stumbled across a way of helping treatments for cancer. That is what it's about. It's about curiosity-driven research and being open to having the vision that from what we learn, we could create new medicines. For me, all of this, the journey and the facts that we learn about how the human body works, It lifts us out of the humdrum of our everyday lives. It's our escape by ascent. Thank you for listening.